Today, I would like to highlight the essential connection between the genetics, lipid metabolism, and the cardiovascular health. So genetic variations, or known as polymorphisms, it can affect how our bodies process lipids, and it will affect the tendency of us in getting the cardiovascular diseases or CVDs. So the three main genes involved in the lipid metabolism and regulation are the APOE, PPAR, and fats genes. CVDs are the main cause of mortality globally, which represents almost 32% of all global mortality. And in year 2020, there was almost 67.3% of all CVDs are attributable to atherosclerotic cardiovascular disease. So next, in Malaysia, based on NHMS, the prevalence of cardiovascular risk factors like the hypertension, high cholesterol, diabetes, obesity are increasing, with dyslipidemia is one of the major risk factors for CVDs. And there were 64% and 56.7% of Malaysian adults, they had elevated total cholesterol and LDL cholesterol level. So the elevation in LDL cholesterol is the key aspect in dyslipidemia, and this is related with the increased risk of cardiovascular disease, especially in the atherosclerotic cardiovascular diseases.
Cardiovascular disease, or CVD, is one of the leading causes of death worldwide. One of the key risk factors for CVD is abnormal lipid metabolism. While diet plays an important role in managing lipid levels, people respond differently to the same type of diet. This variation is often linked to differences in their genes. This study focused on three important genes: APOE, PPAR, and FATs. These genes are known to play a significant role in how our body processes fats. Variations or polymorphisms in these genes can affect how efficiently we metabolize lipids and how likely we are to develop heart disease. The purpose of this research is to identify this genetic polymorphism, understand how they influence lipid metabolism and cardiovascular risk, and analyze how diet interacts with these genetic factors.
I will present about the nutrient role and relationship to disease mechanisms. So first is bad fats, which is saturated and trans fat. Increasing in LDL will cause atherosclerosis, which will directly increase the heart attack and stroke risk. The second is high in sugar and refined carbohydrates, which is this will raise the triglyceride level, which will lower the HDL, which is good cholesterol, which will contribute to the unhealthy lipid profile. The third is good fats, which is monounsaturated and omega-3, which will lower the triglyceride level and reduce inflammation and protect the heart from CVD, which is cardiovascular disease. The fourth one is soluble fiber, which will lower the cholesterol by reducing absorption in gut. So healthy diet helps prevent heart disease by controlling cholesterol, inflammation, and blood sugar. So the second thing is genetic risk factor, which is apolipoprotein E, APOE gene, which it has three isoform, which is E2, E3, and E4. So this E4 allele will cause CVD, cardiovascular disease, and high level of LDL, which is bad cholesterol. Next, second one is peroxisome polyphosphate activated receptor, PPAR, which is control the regulation of lipid uptakes. So the third one is fatty acid desaturase enzyme gene, which is FADS1 and FADS2 gene, which will transforming dietary omega-6 and omega-3 fatty acid into their long chain inactive form. Some mutation will cause poor conversion of some substances to another substances. So this will cause inflammation, and of course it will decrease the heart disease risk.
Today I will share about the methodology or tools and application of nutrigenomics, nutrigenetics, and precision nutrition. These tools help us to understand how our genes affect how we process fats and how this can help to prevent heart disease. First, we start with genotyping and SNP analysis. SNP stands for single nucleotide polymorphism, which is small changes in our DNA that can affect nutrient response. We usually look at three main genes, which is APOE, PPAR, and FADS1 and FADS2. Genes detected using PCR and DNA sequencing. Next, nutritional assessment using 24-hour recall or food frequency questionnaires. This helps estimate intake of omega-3, omega-6, and saturated fats. Test biochemical markers are also used, such as cholesterol level, HDL and LDL, triglyceride, inflammation markers like CRP, IL-6, and TNF-alpha. Then, data analysis tools like SPSS or GWAS, which stand for Genome-Wide Association Studies, to study the link between genes, diet, and health. For the application, first, personalized diet advice. For example, people with APOE epsilon 4 are more sensitive to saturated fats, so reducing foods like santan or butter can help lower LDL cholesterol. Second, gene-based supplements. People with certain fats variants can convert plant omega-3 (ALA) into EPA and DHA, so fish oil works better for them. Third, functional foods like omega-3 enriched eggs or PUFA-rich oil can be matched with someone's gene type. Fourth, we can detect risk early. If someone is more sensitive to fat, we can advise them earlier. Fifth, cultural adapted plan in Malaysia. This means reducing high-fat food like santan dishes or fried foods for those at higher risk genetic. And finally, genomics helps build better public health guideline by combining gene and diet data. So nutrigenomics actually use genetic information to give more personalized diet advice, especially for fat metabolism and heart disease prevention.
We need a personalized prevention strategy to reduce the CVD risk. First one is for EPO carrier. They need to follow a low saturated fat, specifically Mediterranean diet, because Mediterranean diet has high intake of olive oil, fruits, and whole grains, which can help to lower LDL and inflammation. If you have PPAR gamma, you need to choose a high omega-3, high fiber, or low glycemic diet to support insulin and lipid control. Meanwhile, for FADS half variants, you need to boost your intake of DHA and EPA from fish or supplement for anti-inflammatory benefits. And don't forget to combine these diets with regular exercise, stress management, and no smoking for an maximal outcome for reducing the CVD risk. For the treatment, we use gene-based because it will help change how we manage our heart health. For APO carrier, statins may be less effective, so doctor may prescribe ezetimibe or protein converter substitution gexin type 9 inhibitor combined with a healthy diet for better results. For PPAR gene variants, there will be benefit from medications like fibrates, pioglitazone, which improve insulin sensitivity and lipid control, especially in diabetes. For FADS1 variants, taking DHA and EPA supplements helps them to lower triglycerides and inflammation. With genetic testing, treatments can now be more precise, but expert guidance is still essential to make it work.
I will be presenting two major controversies in the nutrigenetics of lipid metabolism along with two key ethical and social concerns. First controversy is research findings are often inconsistent. Genes like ApoE, fats, and PPAR may affect how we process fats, but results differ across studies. That is because other factors such as diet, gut microbiota, and lifestyle also play an important role. So even with the same gene, two people might react to fats very differently. Second controversy is genetic test results can be interpreted differently by different companies. One might say to completely avoid saturated fats, while another recommends only a slight reduction. This inconsistency can create confusion and may lead people to make diet changes based on unclear or unreliable guidance. Next is about two key ethical and social concerns. The first is access. Since genetic testing and personalized nutrition services can be expensive, people from lower income backgrounds may be excluded from the benefits. This could make health inequalities even worse. The second concern is privacy. DNA data might be stored or shared, and without strong protections, there is a risk it could be misused by employer or insurer when making decisions.
In the conclusion, the genetic variation in APOE, PPAR, and FADS influence the lipid metabolism and cardiovascular disease risks. Next is Nutrigenetics and Nutrigenomics enable the development of personalized nutrient strategies. And then the tools such as SNP genotyping, nutritional assessment, and data analysis support precision approaches. Last but not least, precision nutrient has strong potential to transform public health strategy for managing cardiovascular disease, especially in Malaysia. Thank you.